So last time we got as far as delivering the filtrate to the distal convoluted tubule and began talking about the idea that this was the last place where we could reabsorb sodium and sodium ions and chloride ions. Now, the truth of the matter is it's not quite as clean cut as we're going to talk about today. We're going to talk about the idea that ions are reabsorbed in the distal convoluted tubule. That's true. And that the water is reabsorbed in the collecting duct, and that's true too. But the exact boundaries between these two get a little bit hazy. But I think just for trying to keep our thinking in order as we're moving through the nephron and the collecting duct, trying to decide what happens at each of these stations, it's just a lot easier to keep these boundaries more clearly delineated so that the distal convoluted tubule is involved with ion uptake and the collecting duct is involved with water uptake, and we'll work our way through that. So we'll first talk about reabsorption in the distal convoluted tubule. Much of last time we emphasized the role of the distal convoluted tubule and sodium chloride concentrations in the regulation of tubuloglomerular feedback. We're going to extend this one step farther here and talk about the reabsorption of sodium in the distal convoluted tubule and how its reabsorption is under the regulation of the steroid hormone aldosterone. Aldosterone is a steroid hormone secreted from the adrenal cortex. It goes out into the blood vascular compartment and then binds to cytoplasmic receptors in the epithelial cells of the distal convoluted tubule. The binding of that hormone, the steroid hormone, to the cytoplasmic receptors then turns on genes because, after all, as a steroid hormone, it is a kind of transcription factor. So the binding of the hormone to the receptor generates a transcription factor which will now enter the nucleus, and in the nucleus turn on genes. The specific genes that are going to be turned on out here in the distal convoluted tubule will be for so-called epithelial sodium channels. We're going to place these epithelial sodium channels in the apical membrane as transmembrane proteins. We'll recognize here that even though we have reduced the tubular concentration of sodium down around 50 millimolar, we still have about 10 millimolar sodium inside the epithelial cell, so we still have about a 5-fold gradient, a good enough gradient that we can use these sodium channels to allow the sodium to re-enter the cell. Then as those cells begin to fill with sodium, the other player that's invoked by the gene transcription and translation is the sodium-potassium ATPase. So we'll put the sodium-potassium ATPase in the basal lateral surface and pump the sodium out of the cell, and it's going to then return to a peritubular capillary. Just as we saw last time, or time before, that there's a peritubular capillary snaking its way around the proximal convoluted tubule, we similarly see a peritubular capillary snaking its way around the distal convoluted tubule. And under these circumstances, this is where we're going to be taking sodium back up from the epithelial cells of the distal convoluted tubule into the extracellular space and back into the peritubular capillary. Now, for most of our history, this was an important pathway. For most of our history, we were limited in the amount of sodium that was in our diet. But for good reason, sodium is mighty tasty. And so most of us in the Western world, or in the developed world, whatever you want to call it, we, in fact, like to sprinkle our food with salt because it really tastes good. And so if you start measuring the amount of sodium most people are taking in, it's way, way more than they need. And so most of us are not limited by the amount of sodium in the diet, so we really don't do much to invoke this pathway. But for most of our history, we in fact spent a lot of time invoking this pathway to scavenge or reabsorb as much sodium as possible. But in this day and age, not quite so much. There's another hormonal system, a quasi-hormonal system, I want to talk about here as part of the juxtaglomerular apparatus. Now we're going to see here, again, back to the structure of the afferent and efferent arterioles feeding into the glomerulus. Last time we emphasized the story of tubuloglomerular feedback and the release of adenosine from those macula densa cells. Today what we're going to do is try to understand a more extreme case. Now, I'm going to explain this as an extreme case. I'm going to explain this as what happens when there's hypovolemic shock, when there's been some sort of massive hemorrhage and a loss of fluid from the blood vascular compartment. And it works that way. It's certainly important there. But you have to recognize that if you begin to drill down here, this renin pathway is also important in everyday life too. But I think it's most easily understood, and the feedback mechanisms are most easily understood, if we take the extreme case where the renin-angiotensinogen pathway plays a really important role, where there's hypovolemic shock. We're going to see that there are cells located on the afferent arteriole. These cells on the afferent arteriole are known as juxtaglomerular cells. These juxtaglomerular cells monitor for the amount of stretch in the afferent arteriole. And if there's enormous blood loss, then there's a loss of volume in the closed system of the blood vascular compartment, then that stretch will be less. Simply isn't as much fluid inside the tube. And that loss of stretch is recognized by these juxtaglomerular cells. And in that way, the juxtaglomerular cells are saying there's too little volume in the blood vascular compartment. So what the juxtaglomerular cells do is they now release this molecule called renin. Now, renin is not a hormone. All renin is an enzyme. It's in fact a protease, a proteolytic enzyme. And what this proteolytic enzyme does is it binds specifically to a plasma protein called angiotensinogen. This suffix of OGEN tells you that angiotensinogen is an inactive precursor. It in itself doesn't do anything, but this inactive precursor is now activated by the proteolytic enzyme renin. And the proteolytic enzyme, by binding to and clipping out a few amino acids from the angiotensinogen, it now converts that molecule to something known as angiotensin 1. Angiotensin 1 isn't active either. It doesn't do much of anything. But then angiotensin 1 is converted to angiotensin 2. There's this important enzyme called angiotensin converting enzyme that's located on the apical sides of endothelial cells throughout the blood vascular compartment. We associate these angiotensin converting enzymes mostly with the endothelial cells of the pulmonary capillary beds. It's probably simply because we have such an enormous area of capillaries in the pulmonary capillary beds that's where we find most of this angiotensin converting enzyme, often known as ACE. What ACE does is it converts angiotensin 1 into angiotensin 2, and this is now the active player. Angiotensin 2 is the active molecule, and by the time we clip out these last couple of amino acids from angiotensin 1 to make angiotensin 2, that angiotensin 2 now can bind to receptors on arteriolar smooth muscle. The effect of angiotensin 2 bind into these receptors on arteriolar smooth muscle is to cause these muscles to contract. If the arteriolar smooth muscle contracts, what's that going to do to the overall volume of the blood vascular compartment? It's going to make it smaller. Basically scrunch down on the whole thing to make it smaller. So that will help compensate for the loss of blood in hypovolemic shock. That signal then of the renin pathway, or angiotensin pathway, will also impinge upon neurons in the brainstem, or particularly in the, not in the brainstem, will impinge upon neurons in the hypothalamus. And in the hypothalamus, these molecules of angiotensin 1 will bind, these molecules of angiotensin 2 will bind to neurons in the hypothalamus and signal other neurons in the posterior pituitary to release vasopressin. We've talked about vasopressin. Vasopressin is one of these peptide hormones. It's a peptide hormone that regulates the concentration of water in the blood vascular compartment. The way the vasopressin does its regulation is vasopressin, which is released from the anterior pituitary, will now bind to the basolateral surface of epithelial cells in the collecting duct. When the hormone binds to the G protein coupled receptor, it increases vesicular exocytosis. These vesicles in the epithelial cells of the collecting duct contain this transmembrane protein known as aquaporin 2. So fundamentally what we're doing is we're putting water channels into the apical membranes of these duct epithelial cells. Now you remember every time we've drawn the collecting duct, we've always emphasized that it's a continuous tube that extends from the cortex all the way across the medulla and into the renal calyx. So all along that stretch from the cortex into the medulla, the epithelial cells are continuously exposed to an ever higher osmotic concentration in the interstitial space. So now we're going to use the hormone of vasopressin to bind to the basolateral side, increasing the generation of aquaporin 2, and now that's going to provide a pathway for water to run down its concentration gradient from the lumen of the tube into the interstitial space, taking water out of the blood vascular compartment and putting it back into the interstitial space. So this is what's going to occur when we're short water, when we're really thirsty, when we simply don't have enough fluid in the blood vascular compartment. The other side of the coin is when we drink too much water, we've had too much water intake, then what we'll do is we'll throttle back the production of vasopressin. We'll reduce the number of aquaporins in the epithelial cells of the collecting duct. A lack of aquaporins means we no longer have water pathways into that interstitial space, and what we'll do then is we'll retain the water inside the tube of the collecting duct. It will go out into the renal calyx, and from there through the ureters to the bladder, to the urethra, to the outside world. So we'll talk our way through that, and then finish with a discussion of something called the Vasa Recta. The Vasa Recta is an extension of the efferent arteriole. Just like the peritubular capillary is an extension of the efferent arteriole, the Vasa Recta is also an extension of the efferent arteriole, but now this blood vessel is going to run in parallel with the nephron. As it runs in parallel with the nephron, the blood in this blood vessel, the Vasa Recta, is going to come to equilibrium with the extracellular environment. So as the blood flows through the Vasa Recta, it gets deeper and deeper in the medulla. The concentration of urea and salt is going to come to equilibrium with the blood. But then as it runs by the hairpin turn and goes up the other direction, it's now going to get more and more dilute. What the Vasa Recta does is the Vasa Recta maintains the corticomedullary gradient. If you block blood flow in the Vasa Recta, the corticomedullary gradient breaks down. Now it's true that it's the juxtamedullary nephron that forms the gradient, but without the assistance of the Vasa Recta, that gradient can't be maintained. So the Vasa Recta maintains the gradient, which has been generated by the juxtamedullary nephron. And you had a question.
When we talk about the aquaporins and the gut, is that mechanism, is that what affects the color of your urine?
Okay, so the question of color in the urine. And the short answer to what you say is yes. If you have more aquaporins and you're removing more water from the tubule back into the blood, back into the interstitial space, back to the blood vascular compartment, that will concentrate the urine and make it darker in color. That's true. But the darkness in color is in fact a waste product from the breakdown of hemoglobin. So remember, we're turning over hemoglobin all the time, and we have that porphyrin ring that's a waste product. Now, much of that goes out through the liver and generates the bilirubin and biliverdin that gives that sort of chartreuse color to the bile. But some of it, I think it's about 20 or 30%, doesn't go out that way, but rather goes out to the kidneys. And when it comes to the kidneys, it comes into the whole tubular system we talk about. And it's not easily passed back into the interstitial space, so it stays in the fluid. So because it's staying in the fluid and you're moving water from the tube out in the interstitial space, that color gets concentrated. But I don't want you to leave you with the idea that the color of urine is yellow for some reason of the other constituents. It's not from a constituent we've talked about. It's from actually the breakdown products of hemoglobin. Anything else? Yeah.
I'm just curious what that final thing says before exchange.
Counter-current exchange. We're going to see that the blood flow in the Vasa Recta runs opposite to the flow of the filtrate in the tubule, and we'll see that exchange becomes important. Anything else? Well, if not, let's start here with hormonal control of sodium reabsorption. In the distal convoluted tubule. Well, let's draw quickly here the epithelial cells of the distal convoluted tubule. To sort of orient us here, let's extend this over here a little bit. So that this is going to be the ascending limb. This is going to be the distal convoluted tubule. And it's going to then empty into this structure out here. Which again will be made up of these epithelial cells. This structure is going to be the collecting duct. And to remember, the flow of fluid here is from the ascending limb through the distal convoluted tubule and then emptying into the collecting duct. We said that from the story of the tubular glomerular feedback that the concentration of ions here in the distal convoluted tubule are about 50 millimolar sodium and about 50 millimolar chloride. That this was hypotonic to the extracellular fluid, which could be in the neighborhood of 300 milliosmoles. In fact, typically is. If we look at the cytoplasmic concentration of sodium inside these cells, it's in the neighborhood of about 10 millimolar sodium. So now if we wanted to reclaim as much of this sodium arriving here in the distal convoluted tubule as possible, one of the places we'd simply start would be to put in transmembrane protein channels here. We could simply put in these membranes sodium channels. And we do this, and they have a particular name. They're called epithelial sodium channels. And we abbreviate these epithelial sodium channels as ENaCs. In an interesting twist, it turns out that these epithelial sodium channels are also found in taste buds. And so when you taste something salty, it's actually the sodium coming into your taste buds to depolarize those taste buds coming through these epithelial sodium channels. But what we're doing here is we're using this sodium gradient to allow the sodium ions to now leave the tubule and go into the cell. And then on the basolateral surface of these cells will be the sodium-potassium ATPase. And to emphasize then, sort of snaking around the distal convoluted tubule here is an extension of the efferent arteriole. And this is intended to represent the peritubular capillary. So what we're doing here now is we're allowing for the ENaCs and the sodium-potassium pumps to scavenge as much of the sodium as possible, moving it into the epithelial cells, passing it out to the extracellular environment, and then allowing for the sodium to return back to the blood vascular compartment. Now, to be sure, this is a system that's really important to us to get rid of, to glom onto the last bit of sodium. But also, to be sure, for most of us with way more sodium in our diet than we need, this is not something we turn on regularly. But let me tell you the story of what would happen for most of the history of all of our ancestors, where we simply didn't have enough sodium in the diet. We're always running a little bit short of sodium. So we find that the epithelial cells, we can spell that out here, epithelial cells, the epithelial cells of the distal convoluted tubule contain cytoplasmic receptors for aldosterone. Sometimes you'll see your aldosterone referred to as a mineralocorticoid. Aldosterone is in fact released, secreted from the adrenal cortex. And as such, it's a steroid hormone. What the steroid hormone will do, let me sort of blow this up and say this is a distal convoluted tubule cell. And that here in the cytoplasm is going to be one of these steroid hormone receptors. We won't go through all the details here, but recognize that what can happen is that out here near the C terminus, we bind hormone. In this case, this hormone receptor is specific for the hormone aldosterone. When the aldosterone binds to this receptor, it goes through that sequence of events where the binding of the hormone turns on the nuclear access domain, turns on the DNA access domain, turns on the transactivation domain. But the long and the short of it is that this molecule is headed into the nucleus. And in the nucleus, aldosterone, the aldosterone bound to its receptor upregulates gene transcription. No surprise. The genes that are going to be upregulated by aldosterone here in the epithelial cells of the distal convoluted tubule are going to be for the apical sodium channels and for the sodium-potassium ATPases, because these are the molecules, these are the proteins that are involved in reabsorbing the sodium, so these are the ones we gin up in cases where we want to reclaim as much sodium as possible. If we have more than enough sodium in our diet, then we just won't upregulate these hormones nearly as, we won't upregulate the ENaCs and the sodium potassium ATPases because we simply won't generate as much aldosterone. With less aldosterone, then this sodium fluid can move right through the distal convoluted tubules and into the collecting duct into the outside world. So we have sort of this balance where if we have lots of aldosterone, we're going to reclaim lots of sodium. If we have only a little bit of aldosterone, then we won't upregulate many sodium channels. We'll have a dearth of sodium channels out here. The sodium will pass through and go to the outside world. Yes?
How long does the process take to increase the number of channels?
So the question here is, how long does it take from the time that we want to regulate the sodium channels, turn on the aldosterone and get an effect? Minutes to hours, usually up to a few hours. It's one of the reasons why when you go out and have a really salty meal, it's not unusual to get some swelling, because one of the ideas here is wherever sodium goes, water follows. So now you have a really salty meal, and under these circumstances, you have a background of these ENaCs. And so you take the sodium up, you pass it into the blood vascular compartment, water follows, and then you have a higher blood pressure, and you get more fluid buildup out in your limbs. So, but it takes a little while where the system says, we've got too much sodium here. And so what it does then is it throttles back the generation of aldosterone. We reduce the number of ENaCs. We've got fewer aldosterone molecules around. We allow the sodium to then pass through and go to the outside world. And as the sodium leaves, water also leaves. And so blood pressure comes back down, and we no longer have so much fluid out in our periphery. Yes?
In bread, is it bound to a receptor?
So what I'm trying to say here is this orange is meant to be the aldosterone receptor. And then the red overlaying on it is to put the hormone toward the carboxy end of the hormone receptor, because that's where it is. You remember from our stories on the cytoplasmic receptors earlier on, it binds toward the carboxy end. That then signals this whole receptor hormone complex to find its way into the nucleus. And once in the nucleus, it seeks out and binds to the regions of the DNA that are responsible for synthesizing the ENaCs and synthesizing the sodium potassium ATPases, so that you're now more effective. You have more machinery out here to take the sodium up, bring it across the epithelium, and back into the blood vascular compartment. So, did you get it squared? All right. All right. Yes?
If the aldosterone receptor was blocked for some reason, like a drug or something, would that decrease your blood pressure?
If the aldosterone receptor were blocked, would it decrease your blood pressure? Yes, probably a bit. We don't use it that way, partly because it's a pretty delicate balance. Yeah, better not. One of the issues here, sometimes there will be a problem with the adrenal cortex. And the adrenal cortex will stop making aldosterone. And then that's usually called Addison's disease. And Addison's disease is something that John Kennedy had. And if you ever look at the pictures just before his assassination, he's really puffy. And one of the reasons he's puffy is they were giving him enormous amounts of cortisone. And the cortisone then spilled over into the aldosterone pathway, and it then put lots of sodium channels out here in his apical membranes. He was basically taking a lot of sodium back up. Water was following, and that's why he was so puffy. Probably had high blood pressure too, I would imagine. Anything else? Okay, so that's the story of the hormonal control of the epithelial cells and the distal convoluted tubule. But now what we want to think about is the hormonal control of the next set of cells here in the collecting duct. Let's lay this out again. Oh no, before we do that, let's take this, before we go to that, something else I have to tell you about. This is going to be the renin, angiotensin pathway. in hypovolemic. Shock. Well, let's unpack this a little bit. Hypovolemic shock is simply where you have a hemorrhage and you lose a lot of blood. Typically, we can lose a pint or so. It doesn't make that much difference. If you start losing much more of that, you're now reducing substantially the volume of fluid in the blood vascular compartment. And we then have these hormonal mechanisms to respond to the loss of volume in the blood vascular compartment. And the one I'm going to tell you about is this so-called renin-angiotensin pathway. Now, to be sure, if you take another course in physiology, you're going to spend a lot of time in this pathway because it's not only responsible under these extreme circumstances of hypovolemic shock. It also occurs in your everyday life, and you're regulating this release just to control background blood pressure. But let's think about it under the extreme, because I think that's the easiest way to recognize its function and sort out what it does, and talk a little bit about what it can do clinically. We'll begin here with a structure we've talked about a whole bunch of times. Where this is the afferent arteriole. This is the efferent arteriole. And this is meant to be the glomerular capillary, as we've described before, and it's nestled in Bowman's capsule. We talked about this interface last time, and last time we talked about the interface with tubuloglomerular feedback, and in that case, we were talking about the macula densa cells releasing adenosine to regulate the diameter of the afferent arterioles. Well, we're going to add on to that today. We're going to add a new set of cells in here called juxta glomerular cells. Juxtaglomerular cells are modified cells that reside right here in the afferent arteriole. And these juxtaglomerular cells monitor for stretch in the afferent arteriole. So now let's imagine the case we laid out where we have hypovolemic shock. There's been a terrible accident, you've lost a lot of blood volume. If you've reduced the blood volume, what's that going to do to the volume of this afferent arteriole? It's going to reduce it. If it shrinks, what's that going to do to the tension on the wall? It's going to reduce it. If we have a reduction in the tension on the wall, these juxtaglomerular cells say there must have been a loss of blood. They don't know there's loss of blood, they just know that the tension's lower. So what they do is these cells then secrete a molecule called renin. So another way of saying this is the juxtaglomerular cells, in response to a lowered blood vascular volume because of a lack of stretch, release this molecule renin. That's the beginning of the pathway. So let me put this in a numbered set. So 1, renin released from juxtaglomerular cells. 2, renin is a protease whose target is angiotensinogen. Again, OGEN. This tells us it's an active precursor. So renin, released from the juxtaglomerular cells, binds to angiotensinogen. When it binds to angiotensinogen, the third step, renin converts angiotensinogen to angiotensin. One. So basically, renin is nothing more than an enzyme. It's a proteolytic enzyme, and it finds just the right spot on the angiotensinogen, clips out the peptide, and the loss of the peptide from angiotensinogen converts it into angiotensin 1. Then there's an enzyme called angiotensin converting enzyme on apical surfaces of endothelial cells. We abbreviate this as ACE, angiotensin converting enzyme. What ACE does is ACE converts angiotensin 1 to angiotensin 2. Again, it too is basically a proteolytic enzyme. It only cleaves out a couple of amino acids. But angiotensin 2 is active. None of the rest of these function in the blood vascular compartment except to be there. What I mean by that is the renin converts angiotensinogen, which doesn't do anything in itself, to angiotensin 1. Angiotensin 1 doesn't do anything in itself until it's converted to angiotensin 2 by the angiotensin converting enzyme. And this is the active player. Angiotensin 2 binds to smooth muscle, not only here, but more importantly, it binds to smooth muscle, arteriolar, smooth muscle, throughout the blood vascular compartment. So now let's start all over again. Remember, we said that there was a tremendous loss of blood. There was a loss of tension or stretch in the wall of the afferent arteriole. That released renin. The renin then converted angiotensinogen to angiotensin 1. The angiotensin converting enzyme on all endothelial cells throughout the body converted angiotensin 1 into angiotensin 2. Now the angiotensin 2 plays this role in all smooth muscle, constricting the smooth muscle and thereby decreasing the volume of the blood vascular compartment so that whatever blood is still in there takes up less space. And so we basically squish the muscles around the available blood to increase and maintain blood pressure. It's one of the things it does. Now, having said that, and I think it's easiest to think about this under these extreme circumstances, there still is, day in and day out, a role of this system. And in fact, one of the earliest of all high blood pressure medications was a medication they found all the way back in the 1930s that would inhibit angiotensin converting enzyme. So they would give people with high blood pressure this inhibitor of the angiotensin converting enzyme so they couldn't convert angiotensin 1 into angiotensin 2. Without angiotensin 2, there was less effect on the smooth muscle, so there was less constriction, so that provided for lower blood pressure. more recently what we do, although we still use that, more recently what we do is we'll basically lower sodium concentrations by blocking the sodium potassium 2 chloride transporter so that we're not returning the sodium in, so they're not pumping the sodium out of here and putting it back in the blood. We allow it to spill over here, and then we'll overwhelm these ENaCs and potassium channels and spill it out to the collecting duct and go to the outside world. So the sodium potassium 2 chloride inhibitors called Lasix or furosemide then will act as a diuretic in that way. We just spill it out to the outside world. But recognize from this that we do have this effect on the blood pressure. There's one other piece I want to talk about, though, and that's what's happening here in the collecting duct. Because now we have ions and perhaps even some urea spilling over into that structure. Now I'm going to go back up to the upper panel and try again to place the components of the nephron and the collecting duct in position within the kidney. And we've done this a couple of times. It doesn't hurt to do it again, where we'll see now, our emphasis today will be to describe the position of the collecting duct in the kidney with respect to the corticomedullary gradient. So let's redraw that, describing the kidney. Where this is the renal calyx draining into the ureter, and that's going to go to the bladder. And we emphasized some time back that there was this so-called corticomedullary gradient, where out here in the cortex, the concentration of ions was in the neighborhood of about 300 milliosmoles. And you remember that we said under extreme circumstances, down here, deep in the medulla, we can get concentrations here in the neighborhood of 1,200 milliosmoles. The osmolarity down here is derived from about 600 millimolar sodium ions. From about 300 millimolar sodium ions, about 300 millimolar chloride ions, and about 600 millimolar urea. And if you add this up, this comes out to 1,200. So the concentrations here are around 1,200 milliosmoles. Now, let's lay out the nephron and the collecting duct in this system. So there's the proximal convoluted tubule, Bowman's capsule, proximal convoluted tubule, descending limb of the loop of the nephron, the hairpin turn, the ascending limb out here to the distal convoluted tubule. And that's where we've been today. We've been talking about the epithelial cells there. But the distal convoluted tubule then empties into the structure I'll put in red, called the collecting duct. Now, as I have implied here, as we've talked about in the upper panel, this collecting duct is simply, again, a tube constructed from epithelial cells stacked one next to another, all along the length of this collecting duct tube. But one of the things to recognize is that as the fluid spills over, as the filtrate spills over from the distal convoluted tubule and enters the collecting duct, it now flows through the collecting duct and the epithelial cells along the collecting duct are facing an ever higher interstitial concentration of ions and small molecules. So all along this collecting duct, these epithelial cells are facing an ever higher concentration till down here deep in the medulla, concentrations approach 1200 milliosmoles. So on the right-hand panel here, let's now talk about the regulation of water reabsorption. Talk about the regulation of water reabsorption in the collecting duct. Now let's take an extreme example again. One where you haven't really had anything to drink for a couple of days. So you're really, really thirsty. So under circumstances where you're exceptionally thirsty, what do you want to do? You want to retain as much water as possible. So what happens is that up here in the hypothalamus, there are cells, there are neurons that monitor blood osmolality. And so what's happening here is these cells are saying, it's getting salty in here. And so what they do is they signal now other cells here in the posterior pituitary. When these cells signal the cells in the posterior pituitary, these release a peptide hormone called vasopressin. Now, in these epithelial cells, I'll label this as a collecting duct. Epithelial cell. And again, to orient you here on the basal lamina, on the basal lamina, on the basilar side, there will be these transmembrane proteins, 1, 2. Again, a G protein coupled receptor. And this G protein coupled receptor binds to vasopressin. Now you remember that one of the things that hormones can do is they can regulate exocytosis and endocytosis. So now under circumstances where we are really thirsty and we're generating a lot of this vasopressin, we're now inserting into the membrane here these vesicles which contain aquaporins. Abbreviated AQP2. So now into the apical side of these membranes we're putting water channels. And look what we have in the basolateral side. Out in the basolateral side, we have then osmolality that may be as high as 1200 milliosmoles. So now the water, which may be up here in high concentration because we're down to around 50 millimolar sodium and chloride, now we have lots of water in here. And now that water can move across the corticomedullary gradient